Okay, great. The meeting is recorded, so don't worry if there is some terms in the title that you don't understand. I will try to explain what I'm doing, and I will try to do so for a large audience. So first of all, as Karin mentioned, I'm an associate professor at Linköping University, but I'm working on the Norrköping campus that is in Linköping University. And here you can see a short movie that shows a bit the campus that is around the river. And here you can see the visualization center. So this is where I work. Here, this is the visualization center. And on the other side of the river, this is where I spend also 50% of my time in the laboratory of organic electronics. And I'm also associated with the Swedish e-science research center that aims at promoting e-science, so which is the field of research that is connected with supercomputing mainly. So I'm a theoretical chemist. So what is theoretical and computational chemistry? So theoretical chemistry, this is a branch of chemistry which develops theoretical generalization that are part of the theoretical arsenal of modern chemistry. So for example, we study the chemical bonding, we study chemical reaction, we study how the energy of a molecule is connected to its conformation, which is called the potential energy surface, and also the electronic structure of molecule, so which is connected to what we talk about orbitals of molecules, and so on and so forth. And what is computational chemistry? Well, that's basically the implementation of theoretical chemistry method on supercomputer. So here you can see a picture of a supercomputer on the top right. So this is the one at KTH, but in Sweden we have many different supercomputer and also in Linköping. And those supercomputer allow us to do calculation in a day that would take 20 years on your laptop, actually. So in the arsenal of theoretical chemistry, well, we have a lot of different methods, and those methods, they apply at different, for systems at different scales and for different time. So if we want to study very large system, we're going to use Newton mechanics. And if we want to study small system, we're going to use quantum chemistry. So let me go briefly about those two approaches. So for very large system that we can study for a long time, we're going to use Newtonian mechanics. So you're all familiar with this, I'm pretty sure. This is the famous equation, second law of motion of Newton here. And we're going to apply that to the atoms. And we're going to be able like that to study the atomic structure. So how the molecules are assembled, how they're gonna interact with each other when they are placed in a medium like water, like in solvent, and all that. But when we do that, most of the time we neglect the electrons. So we just consider the nuclei of the molecule. If we want to study the electrons in our system, then we need to use a different approach, which is quantum mechanics that all of you heard about, and the famous Schrödinger equation here. And in this case, then we're going to be able to study not only the nuclei, but also the electrons, and also how those electrons can transition from those, what we call those electronic level. And here we're going to be able to access information such as the absorption and emission of light by molecules. And in between those methods, there is what's called the QMMM method that was awarded the Nobel Prize in 2013. And here the idea is to take the best of both worlds, to combine the QM and the MM, so the quantum mechanics and the molecular mechanics, to be able to tackle a larger system and to be able to gain information on larger and larger systems. And it was awarded the Nobel Prize in 2013 to those three fellows, Karplus, Leavitt, and Warshel. So let's go back now to what I'm going to be talking today, which is molecular mechanics, molecular dynamics, which allow us to study the evolution of a chemical system with time. And when we have our supercomputer and when we study our system with time, this is a bit what our program gives us, right? So here we get, for time 0, we get a list of coordinates. So here you get different atoms and you get a coordinate, X, Y, Z coordinate. So that would be the position of an atom in a Cartesian coordinate here. So that's for the time 0. And then we can propagate this and see how it's going to evolve with time. And this is for a later time. We get another file that tells us what is the position of the atom for this, for the next step, and so on and so forth. But obviously, this is not very user-friendly to watch those files. I mean, to look at those files, and we don't get a lot of information. So what we want to do is to be able to visualize it as a movie, more or less. And so this is where I'm working in, mainly with the visualization, the scientific visualization group, and we developed a software that is called VIMD, which aims at visualize and analyze our molecular dynamic trajectory here. So let's start with a very simple example of a molecular dynamic trajectory, something that you are all familiar with. If you would take a cube of ice and put it on your kitchen counter and wait for it to melt. So here you see water organized in an ice crystal here. And if I calculate how my system is moving, and if I start to increase the temperature in my simulation, so you see that now I have water that is nicely organized in a crystalline way. Those pink bonds here represents what's called hydrogen bond, which is the weak bonds that tie it together. And if I increase the temperature during my simulation, I see that there is some disorder that starts to appear, and it's not staying anymore in this ideal position of the crystal. And if I continue to increase the temperature, well, what I will see is, like, at the end, I'm going to arrive to a completely disordered system, which is now, here we are, which is now the liquid water, which is how the water looks like. So that was first simple example of a molecular dynamic simulation, and I wanted to choose this example because I think that anyone can relate to it. But let's continue a bit the experiment in the kitchen, and now let's say that in your water you're going to make some pasta, right? So, and you all know that when you make some pasta, you need to add some salt in your water. So here, this is a cube of a small cube of salt. So we got the sodium atom in blue and the chlorine atom in green, and they are in water. So here I just represent the water that is around this cube and not the full water that we have in our simulation, because otherwise we wouldn't see anything, right? And then I can see how it's going to evolve with time when I run my simulation. And so this is what we do here. And what you're going to be able to see is that after some time, well, the water is going to dissolve this cube of salt, and you're going to be able to see one atom that's going to detach from this cube and get solvated, and like that leave the cube. And yeah, and I also show here the water around this atom that is moving away. And if I continue my simulation for some time, so here I'm going to speed up a bit the process here, but what you will see is that at the end of my simulation, here I accelerated a bit, you know, but then at the end of my simulation, you can see that I have completely dissolved my salt, my cube of salt here. So that was just two simple example of molecular dynamic simulation, very simple case that I just created for this talk, because
just a quick question.
Yes.
Yes. Can you choose also the temperature of the water if it's cold?
Yes, we can play with different parameters. We can play with the temperature that is maintained by the thermostat. So you're all familiar with the term thermostat, of course. So we can play with the temperature. We can play with a lot of different parameters in our simulation, indeed. For instance, the first simulation that I showed you with the water, I was increasing the temperature during the simulation. And here I didn't do that. It's just a long simulation, but it takes time before the water can dissolve this cube of salt.
And then you can compare how long time it takes when it's cold water and hot water, for example.
Exactly. We can do a lot of different experiments. We also call that experiment, even if it's on computer, and we can do a lot of different experiments like this to model well, what's happening in reality, between brackets. So that's the type of simulation we can do. Here I chose those very simple example that you can all relate to, but now I'm going to switch to one of the research subjects I've been working on for some years. And so it's a much more concrete case, and it's connected to Alzheimer's disease and what's called amyloid disease. So so what are amyloid disease? So they are diseases that are connected with the misfolding and the aggregation of protein in the brain. So there is different, there are several diseases that are connected to this misfold of protein happening in the brain, like Alzheimer's, Parkinson, Creutzfeldt-Jakob, and also the type 2 diabetes. So what's happening is like some protein in the brain have some forms, and there is a misfold happening somehow. We don't know exactly how and why, but they're gonna start to form long filaments like this. And those filaments are gonna aggregate and to form larger and larger structure like this. And the more the disease progress, the larger those aggregates built in the brain. And what would be ideal is to be able to detect those aggregates at an early stage of the disease, to be able to treat it early, actually. So, so if we now focus just on Alzheimer's disease and we look at the brain, well, this is a very schematic picture of the brain. I know that Karin is smiling because she's an expert of the brain, so it must be very ridiculous. But anyway, I'm going on with this image. So there is aggregation that's happening in the emotional brain. That's called the tau aggregation, and some other happening in the thinking brain, the neocortex, are called A beta. And in a healthy brain, those aggregates build, so those aggregation of protein into fibril is going to build, but it's going to be flushed naturally by the brain in a healthy brain. But in a sick brain, those aggregates not going to be flushed, and they're gonna those misfold of protein are going to build up in the brain at the early stage of the disease. And when the disease advances, we're not going to see A beta only in the neocortex and tau in the emotional brain, but we're going to see migration of tau in the neocortex and migration of A beta into the limbic system. So what would be ideal is to be able to detect those misfold of protein and to be able to detect in a selective fashion the tau and the A beta protein in the brain at different stage of the disease, because that would be an indication about how much the disease advanced. So how to do that? One way to do that is to use molecules. Well, because molecules, some specific molecules that are designed by a group in Linköping, by the group of Peter Nilsson, yeah. Some molecules can be designed and can be designed to attach specifically either to tau or to A beta. So here you see one molecule, it's just a name, you don't need to know about the structure of this molecule, it doesn't matter so much for the moment, that emits light, green light, and another one that emits red light. And you can see that you can also mix them together. So when you mix them, well, you see that you have some zones here that are mainly red here, and some zones that are mainly yellow. And it means that this one of the specific molecules is going to attach mainly to tau, and the other one's going to attach mainly to A beta. So what we want to understand is where are those molecules attached to the protein? How do they attach differently to different type of protein, A beta or tau? And so we want to know where are the binding sites, so which means where they're attached. We want to know how the color of the probe that you can see is governed by the conformation, because different conformation of the same molecule is going to emit different, the molecule in a specific conformation is going to emit different light. And this is what we want to calculate and what we want to understand. And we want to understand what is the conformation so of the molecules when they are bound to the protein. And we also want to understand if we can design probes that would be that would bind more efficiently to one protein or another, and also if we can help to design more selective type of probes or molecules.
Mathieu?
So, yes.
Sorry, how do you do to see these molecules?
All right, so how to obtain those nice images that you see. So this is based on, so what is done is like you're gonna inject the molecules of those images, they are fluorescence images, right? And thank you for the question, I should have mentioned that. So what is done is like you will inject, so they are done on mice, those images. So you would inject the molecule in the tail of a mouse, sorry, and then you would well, scalp the mouse and look directly on the living mouse. Look at do an image of the brain of the mouse that is living actually, and do a fluorescence imaging of the mouse.
Okay, so you see the brain of the mouse.
Yes, exactly.
Real, and then you see some part of the brain that are with
yes. You make what's called a cranial window actually. So you just scalp the mouse and you inject those molecules in the tail of the mouse, and those molecules, they pass the brain-blood barrier, and they are found ultimately in the brain, and they're gonna aggregate on the different misfolds, so those tau and A beta protein.
Great.
So this is done, but ultimately the point would be to be able to do it directly on blood sample of patient, obviously, to be able to see those misfolds, because those misfolds, they are also happening in the blood. So that's the long-term plan here. So I've been talking about A beta and tau for a long time, but I didn't really show those misfolds how it is. And for a long time, we didn't have very proper knowledge of the 3D conformation, the geometry of those proteins. But since the advancement of cryo-EM, that was awarded the Nobel Prize in 2017, now we get very nice atomistic information about how those fibril, those long chains of molecule looks like. So here, this is a representation of the A beta protein, and here this is the tau protein. And you see that they look quite a lot alike. They are long fibril. Here, each chain here, they are a chain of amino acids, and they are connected with each other with hydrogen bond, and they form very long assembly of protein like that. And then we have our molecules, and what we want to know is where is the molecule gonna interact, right? Where it's gonna bind at which site it's gonna bind. So what we do in our simulation, so we can use the information that we get from cryo-EM, and this is here an atomistic representation of this tau protein I just showed you before. And then we're going to build a box in our system with this long chain of protein, and all of them, they are repeating unit here. So we get 253 units that repeat each other. In our box, we're going to put those, this assembly of protein. We're going to put number of molecule here 100 molecule, and we're going to put water, of course, because all those happening in the brain, I mean, mainly water, like in our body. And at the end, we get a box that is roughly 3 million atoms. And we can perform simulation, and we model this for a certain time. And just to give you an idea, we do those calculations, of course, on several CPU at the same time. But this type of simulation takes roughly a month, actually, just to give you a rough idea. And at the end, what we have is a movie, as I showed you, of what's happening in this simulation. So we have a lot of information in those movies. We get probably too many information, and we need to be able to concentrate those information into something that would make sense, right? So as I told you, the protein is actually made of 253 times repeating units. So what I can do here in the software that we develop is I can say, okay, since there are repeating units, maybe what I would like to know is I would like to be able to superpose them, right? Because I get a long simulation and they are the same unit that repeat each other, but what I want to do is to be able to superpose it. And that's what I'm doing here. So here I get superposed my 253 chains onto each other. And this is done by the software we develop, right? And so that's the first step. And in the second step, I want to know, okay, where are those molecules interacting with those chains, right? We want to know exactly where they interact. And I can do that, and I can filter here. I can use a filter, and I can say, okay, I want to know where are my res name PFTs, which is my molecule in my system, and I want to be able to compute the density. So here what you see is where my molecules are interacting with my protein. So the red zone shows the high probability of the molecule, right? So that would be here mainly, that's where the molecule is, and the blue zone, so where there is a lower density, and of course when there is white, then there is no probability at all. So here with this program, I can concentrate all this information of my 253 chains and my 100 molecule into one single image that tells me, okay, this is where my molecule is binding to my protein, this site here.
And this site is referring to a place in the brain, or? Is
referring to a place in the misfold of this A beta protein, right? So here I'm not at the brain scale, I'm more at the misfold scale, and I can see where this molecule is binding to my protein.
Okay.
And like that, I can have, so this is here, I get a filtered visualization of my probes, and I can see where they are interacting, and I can get more information, which is, for instance, what is the strength of the interaction, what is the geometry, how is the geometry affected by the fact that it's interacting with my protein, and so on. And I can, like that, detect some specific sites where my molecules are interacting with the protein. So that's how we do it. And when we have that, we have an atomistic information about the specific interaction between the molecule and the probe. And that's for us the first step to be able to study then later what is the emission of light of those molecules, how they're going to emit light. And also this is the first step to think about, okay, how can I now modify my molecule? How can I add a group, for instance, here, or remove a group here and there to strengthen this interaction, or to make it weaker, and so on and so forth. The point is that we have, now I'm just showing you an example with one single molecule, but of course we have a lot of different molecules, and those molecules, they're going to interact competitively between the A beta that I showed you and the other tau protein that I showed you. So what we do is we do simulation for a lot of different molecules with a lot of different misfold of protein, and like that we are able to build a library of molecules that's going to be selective and going to help us in the future to detect better those misfolds for specific protein. So I think that's about it for my talk. So I wanted to just to conclude, so we developed a generic framework that is done for molecular dynamic visualization. I showed you here an example based on this detection of amyloid fibril connected to Alzheimer's disease, but it's used for many other things in the field of molecular dynamic. So it can be material science. Here it was more health, but it can be material science. We develop this interactive definition of properties, and we can aggregate and explore our dynamic, actually. We connect the visualization to the analysis, and it's also used to develop visualization for popular science application, in particular at the Visualization Center in Norrköping. So, and if you don't know about it, I really encourage you to go and visit the Visualization Center. So to finish, I would like to acknowledge the people involved. So in the group of visualization, this is the PhD student Robin that is doing the main development of this software, helped with Martin and supervised by Anders Hinnemann, Ingrid, Hotz, and myself. The experiments that you saw, so this A beta detection, this Alzheimer's disease, is done by the group of Peter Nilsson in Linköping University, and all those spectroscopy that we do, so which is the light matter interaction, so how those molecules absorb and emit light, is done by the group of Mikael Ågren. I would like to thank my group of, so the group of visualization, and the other 50% of my time I'm working in the laboratory of organic electronic in Norrköping. And I would like to thank CERC for funding, and the SNIC, which is the Swedish National Infrastructure for Computing, that allow us to do all those heavy calculation on supercomputer. And I would like to thank you for your attention, and I would be very happy to answer questions.
Great, thank you very much, Mathieu. I don't know if you see, but there is question online.
Yes, I just seeing them.
Okay, so we can begin with if you can answer this question. I don't know. You will tell.
Okay, what molecule is responsible for the symptom, or are those only interacting with each other? So I'm not. I just want to say that I'm not a specialist of Alzheimer's disease, actually, at all. And what is causing the misfold of those proteins, what is causing it or amplifying it and making it worse? Actually, there is a big debate in the literature, and we don't know what it could be. There is a hypothesis that heavy ions, so like heavy metals poisoning, could make it worse. But there is obviously a genetic factor in those diseases. This is well known. But so I'm not a specialist of Alzheimer's disease, and I don't know what is the cause of those misfold of the protein. Actually, that was a question for VHEL. I don't know if I answered the question properly.
I think so.
Okay, how knowing there is another question from Laura Torres Garcia, who's asking how knowing the dynamic of the molecule could help to develop drugs against interaction or aggregation of the molecules. Well, okay, so that's not really what I'm interested in, but I mean, that's not what I've been talking about, but obviously we, in my field, there is also a field that is based, that is called drug design, right? So the idea would be to study how the molecule is interacting with the protein, and like that you can imagine. So, for instance, maybe I can maybe show you something here. If you just give me a second, I'm going to show you. I'm going to show you something. I hope that you can you see anything here on my screen? No?
We see the PowerPoint still.
You still see the PowerPoint, yes. Let's see. I'm going to show you another dynamic of an aspirin molecule that is bound to a protein. So let me see here. So do you see here?
No.
No, you don't see. Okay, I'm not showing the right stuff. So let me see here. Now you can see it, I guess.
Yes.
All right. So this is here. This is a dynamic of this is an aspirin molecule that is bound to a protein, actually. And we can, so I don't know if I get a real movie here, but okay. So we get those type of structure, and we can study the dynamic, how the molecule is moving inside this protein. And with computer simulation, what we can do is we can do substitution, right? So we can add a group, for instance, to this molecule, or remove a group from this molecule, and we can see if we're going to increase or decrease the binding, which is the affinity of this molecule. So this field is called drug design, and those type of simulation are used a lot by the pharmaceutical industry to improve drugs. And obviously there is a huge benefit, because instead of synthesizing, let's say that you have 100 candidates, right? You don't want to be able to synthesize those 100 molecules, and you don't want to test them then on mice. Instead, you can do those type of simulation first to identify potential molecule that's going to be the most efficient. And then you're going to narrow it down to, let's say, 15 molecules only that you would have to synthesize, that you could synthesize and then test in vivo, in vitro first and in vivo then. And that would be a huge gain of time and money, obviously. So those type of simulation, they are used a lot. Molecular dynamic simulation are used a lot by the pharmaceutical industry. And here, the visualization that we are developing are going to be, I hope, also useful in the future to help the analysis of those long trajectory and long dynamics that are performed by the pharmaceutical industry.
Thank you very much, Mathieu. This was very, very interesting.